enzymes from that virus. The enzymes of that virus will degrade the host DNA. Okay, it breaks it up into small pieces. Now, the virus that has infected that bacteria uses the bacteria's cellular machinery to make copies of itself. Okay, so now we're synthesizing new viruses. When these viruses get made inside the host, sometimes a piece of DNA, that host DNA that was degraded, sometimes a piece of that will be trapped into one of those new viruses. Okay, so what happens is those viruses, after they have replicated enough of them, they will burst the cell. We call that lysis. When that cell bursts, that will release all those newly formed viruses. That one virus that trapped part of the DNA from the host will then go and infect another bacteria. We call that one the recipient. When it injects its DNA into the new host, that DNA from the original host, which was contained inside that virus, will now be able to be incorporated into the DNA of the recipient. And what happens when a bacteria brings in DNA and incorporates it into its genome? It's going to be able to express those traits. It will express the traits of the original donor. I know you got a question. Go ahead. No? No question?
No.
None?
Go.
You want to, huh?
Can you go over my bubbles?
Okay. Injects DNA into bacteria's donor or host. You can call it the host. Okay. Causes DNA of host to break up. What does that? It's the enzymes do that. Virus uses bacterial to make copies of itself. When cell breaks up, one that contains original DNA, host DNA. Okay. Host DNA gets trapped inside one phage. So that explains what happens down here. So when the cell breaks up, the one that contains the host DNA may infect, and it will infect a recipient. DNA from original host becomes incorporated into DNA of recipient and is able to express the traits of the original donor. Correct. Moving on. Last one. Huh? Last one is where we explain in detail how bacterial conjugation occurs, and we're going to use the F+ cell model. Now, one thing you have to understand about the F+ cell, bless you, the F+ cell contains an F+ plasma, or F plasmid, okay? On that plasmid, you have an origin of transfer. You have a gene that expresses the making of the pilus. Okay?
Gene that explains the making?
Expresses or...
Gene that expresses.
Okay, yes.
Making of what?
Making the F pilus. The pilus is the projection off of the cell that binds onto another cell to allow the transfer of the plasmid. Okay, so it has to contain those traits, right? the ability to transfer, and the ability to make the outward structure that allows for a transfer, okay? So we have an F plus cell that has an F plasmid in it. You have another cell that is normal, and we will call him the recipient. F plus is the donor. The F plus cell will come into contact with the F minus cell.
Wait, what, where's the F minus cell?
Yeah.
He's the recipient.
So we can just say that the F plus cell comes into
Contact with the F minus, yes, that would be
with the
fine.
recipient?
They're both the same people.
So it doesn't matter what word I use?
No, it does not, because you'll explain that later on at the end of the thing. Anyways, it comes into contact with it. The pilus will attach to the F minus cell or recipient cell and draw it closer to it, okay? Now, at the origin of transfer, part of that plasmid will break off and go through that F pilus into the F minus recipient cell.
Wait. I didn't hear you.
At the origin of transfer, part of one strand of that plasmid will break off and go through the pilus and enter into the F minus or recipient cell. Once that transfer is complete, now you're going to have replication occur, right? So now I have to, because it's two strands, it's DNA, it has to have two strands. One strand went to here, one strand went to there. While they're inside there, they will make two strands. So now you have two complete plasmids. So now, the recipient cell now contains a F plasmid. Since it has an F plasmid, it's going to start expressing the traits of a F plasmid containing cell, which means a pilus will be formed on that new cell, as well as it will also express the traits of anything that came from that donor cell. So now I have two F plus cells. One was F plus, the other one was F minus, but after the transfer, now I have two F plus cells, because the recipient takes on the same traits as the donor.
Yeah.
So now that is horizontal transfer. And, huh? Okay. That's it, that's the last
What
one.
about my shortened version?
What is your shortened version of it?
See if we'll cover it all.
We'll see if that passes
Okay.
muster.
In bacterial conjugation using the F positive cell, the donor cell attaches to a recipient cell, which is F
By way of a
negative,
pilus.
with F negative with its pilus.
Yes.
Cells are drawn together. Donor strand, F positive, is transferred to the receiving
Donor
cell.
strand of what?
Hmm?
Donor strand of what?
The donor strand
Of
of
what?
the F positive.
Plasmid.
Or, okay, the donor strand.
It's not coming from his genomic chromosome, is it?
No, a plasmid is transferred to the receiving cell and becomes a complete plasmid of F positive and expresses the donor traits.
Okay,
That work?
that will work.
Okay. I'll just add the plasmid.
F plasmid and F plus.
I got the F plus and
I
negative.
didn't get that.
Yeah, but at the end now you have two F pluses.
Is it still saying?
Right,
Okay.
becomes a complete plasmid of F positive and expresses the donor traits.
So F I is not positive, so you need the F plus.
Wait,
I'm glad you're giving it credit for becoming
everybody
complete.
else can have.
I did.
So,
So, because I started with an F positive and F negative, and then I ended up with another F positive complete plasmid.
Okay, complete.
I already have it drawn in my notes, actually.
So
Anybody
can
else?
we draw this one on the test?
Let me see. Yeah, as long as you explain it, because I'm reading it as if I have no idea what you're talking about.
I do have a question. On the serial dilution,
Yes.
explaining that as long as we put like a serial dilution is performed when you need to dilute a liquid colony to get a better isolation. And then can we just put it as a series of tenfold dilution?
Tenfold,
Yeah, you know, 1 to 9, 1 to 9, 1 to 9, you still have a tenfold dilution
okay.
with 10
If
milliliters.
you wanna word it that way, sure.
And as you do the liquid dilution, you take a sample and put it on a variable plate. I mean, how are we gonna add in the variable plate now in Word?
Viable plate
Or I
count?
mean, viable plate count. Taking a sample,
Okay, you can take,
Line it.
you can say, for instance, if a plate was, made from this, dilution, I would get, too much growth count. If I took it from this one, I would get too much growth count.
Okay.
If I get a sample from this one, then I would get approximately 65 to 100 colonies, which
Okay.
would still make it difficult to count.
okay,
Otherwise, I could do the last one, and if I was to plate this one, then I would get a viable plate count because 6 to 10 colonies would have been produced.
okay, so like if I was telling you what the serial dilution is.
Yeah.
And,
As if
and
you were-
also utilizing viable plate count if I were to.
Yeah.
Then it would all be covered.
Yes.
Okay, got it.
You know, so as if you were trying to explain it to somebody who didn't know what they were talking about.
All right.
Well, Jay, you should explain to me, because I don't know what you're talking about.
I do explain it to you guys. I explain it in many different ways.
Yeah, I know. Thank you very much.